DNA is derived from a much longer word, deoxyribonucleic acid. The D for the first letter of deoxyribonucleic acid, the N for the nucleic part, because that's one of the places where DNA is, in the nucleus. And notice the similarity between nucleus and nucleic acid, and the A for acid. James Watson and Francis Crick and Maurice Wilkins. And there was a fourth person who really inspired them, but she did not get a Nobel Prize. She died of breast cancer before that happened, and didn't get the credit that she ordinarily would. But here are some young people earning a Nobel Prize, and they're just at the beginning of their careers. Often we think, well, you have to be at the end of your career to do something spectacular like this. But it's interesting, if you suggest a project to a young person, they figure out you can get a result. They do. Now, while this is sometimes referred to as the Watson-Crick structure, what they discovered was that it had to as a double helix, and to describe it, if you think of a stairway that swirls around, on the stair steps, those are what are referred to as bases, and what would be in each stair step would be two bases. There are 4 bases altogether in DNA, and DNA is composed of some subunits referred to as nucleotides. A nucleotide consists of a phosphate, a sugar, and one of 4 bases. The sort of railing, or sort of if you were holding your hands going up a stairway, the borders are alternating phosphates and sugars. Phosphate and sugar, and then phosphate and sugar. And you have 2 of those, of course, if thinking of a railing for both sides of a stairway. And these stair steps then, referred to as bases, there are 4 of them. There is one referred to as adenine, and it's always bound with a second one called thymine. So it could be adenine thymine or thymine adenine, but these are complementary, and they're bonded in such a way that they can at some time be separated by an enzyme. The other 2 bases are guanine and cytosine. And once again, it can be other ways, cytosine or guanine. So the nucleotides then, when they say that it's a phosphate, a sugar, and a base, it could be one of those nucleotides, could be the phosphate, sugar, and a base, and adenine. Or a second one could be a phosphate, a sugar, and a thymine. Or a phosphate and a sugar and a guanine. Or a phosphate, a sugar, and a cytosine. And when these bases are shown in often the literature, what they will do is they ignore or don't bother with the phosphate and the sugar, because that's always there. What they're interested in listing would be the particular base, and they might even do it horizontally, and you can see rows of them. So when you hear that a nucleotide, and I refer to one, it's the base that they're interested in. When you think about it, you perhaps have heard of DNA is a code. It's a code for all organic life. That is a frog or a person, a plant, or something else, but anything that is living. So we have that in common all together, that we have this DNA. Well, if that's what is a code that makes us unique, and it does, how can that be when they're just 4 bases? You know, some cakes have more ingredients than DNA has. So what the answer is why you are unique, or a piece of corn cob is unique in its way, is that it's the sequence of the bases. It's the order of the bases that spells out instructions to the cell. So DNA, one of its features, important, it has the code that gives instructions to the cell, and this sequence of bases that makes you, or any other kind of life, what it is. So that's one of its characteristics then. The DNA is in the nucleus of a cell, but there is another DNA in a cell that's not in the nucleus. And many people are only now becoming aware of it. The area around the nucleus that surrounds the nucleus is called the cytoplasm, and there are substructures in that cytoplasm. Your book illustrates 2 of these because those are the ones that they will be referring to in this book. And one of these is called mitochondria. That's the plural, or mitochondrion. And mitochondria have DNA. They call it mitochondria DNA. And in writing it, they distinguish it from nuclear DNA with a little letter M and small case t, so mtDNA. And this is becoming people more aware of it because you can trace generations through that with that DNA. So it's become quite popular in some TV programs called My Ancestry. There are other names, other programs where people are interested in knowing where they came from geographically and generations back. Mitochondria is what they would check, those DNAs, if the person inquiring is a female. The sperm does not carry cytoplasm. It carries nucleus of the male. So that's why they say, well, everyone gets their cytoplasm from their mother. Looking into ancestry then, the female, if she's going to trace it, it would be through the mitochondria. The male has another choice. They can check ancestry not only through the mitochondria in the cytoplasm, but also through the Y chromosome. And with the Y chromosome, he would be checking his ancestry through his father's family, the mitochondria through his mother's family. So there are these substructures. Another substructure inside the cytoplasm is called ribosomes, and they are very important for the formation of proteins. So we'll get to that a little bit later when we talk about protein synthesis. DNA, besides the code of establishing what type of life you will be, it can replicate itself. So in replicating itself, where you had one at first, when the replication is finished, you have two identical ones. Please take out the part of a DNA molecule so you can see what I will be pointing out to you about it. If you have it out now, what you might notice is that the bases, which are different colors, so you can extinguish one from the other, what I want to call to your attention is the shape. And those bases that are complementary to each other fit together like a jigsaw puzzle would be. The thymine curves outward and the adenine curves inward, so as if you could put two pieces of a jigsaw puzzle together. And that's to communicate to you that those two always are together. And then you look at the thymine. So thymine and adenine are bonded together. You'll also see some little horizontal lines between the two to show that that bonding can easily be separated. It can be separated by an enzyme. So sometimes they're together, but at other times they will separate. At guanine and cytosine, they too are like a jigsaw puzzle, but one has a sharp point and one has sort of a groove that goes inward. So there's no mistake that they are complementary with each other. If you look in the upper right part of the DNA molecule displayed, there's sort of a light golden rectangle that goes over adenine, the attached sugar, and the phosphate. And it's labeled. It says nucleotide. Before the online teaching, it never occurred to me how popular or how frequently the word nucleotide would be used in reference to the virus. It's in the newspaper. Who would think such an obscure word that you find in a textbook? I would hear radio announcers talk about nucleotides, and so in case you read it in some place in the newspaper or elsewhere, you now know that what it is. It's one of these bases, and they're really focusing more on the base, but the whole package is called a nucleotide. So what a surprise that was, that would become a common term during this time. In addition to DNA, there is RNA. It stands for ribonucleic acid. It differs from DNA in that it is one strand, in contrast to DNA's double helix or two strands. It has uracil, shown by the letter of the alphabet U, in place of thymine, which is shown by the letter T, and it also has a different sugar, which why it's called ribonucleic rather than deoxyribonucleic acid. There are three different kinds of RNA. Messenger RNA, shown by a small m in front of RNA. The formation of messenger RNA is referred to as transcription because what it's being transcribed is information from the DNA. DNA then is a pattern or a template upon which messenger RNA is formed. A section of the DNA helix parts, and so each of the strands that are separated are like a pattern, and one of these strands where the code is for messenger RNA will attract free-floating nucleotides to it. Transfer RNA, shown by a small t for transfer in front of the capital letters RNA, and last of all, ribosomal RNA, shown by a small r, RNA. So first of all, featuring messenger RNA, it gets its information from the DNA in the nucleus, so it is in the nucleus as it forms, and it's going to leave the nucleus, go outside it, over to ribosomes, where amino acids are put together to form a protein. And what you need to notice is that while DNA is made up of 4 bases, here amino acids are 3 bases. So when you look at the illustrations, you're going to see things in groups of 3 bases. An amino acid is referred to as having a triplet of bases. In other words, it consists of 3 bases. Proteins are made up of amino acids, so of triplets of 3. And it varies with the protein as to how many of these triplets it takes to bring together a protein. It sometimes was described by one person I compared it to as a child's train set, where you're hooking up more cars together to make the train longer. Well, here it's joining more triplets until you get the specific number that is needed for the formation of a protein.